Hello everyone and welcome to my teaching presentation titled Cell Cycle Regulation in the Cell. My name is Emerildo Souza and this presentation is part of the course CHM 530 Concepts of Biochemistry that is taught at the Grand Canyon University by Professor Dr. Hector Rivera Jaques. In this class, I will be presenting an overview of how the cell cycle is regulated at the molecular level. This topic is a core concept in biochemistry and provides the foundation for understanding normal cell growth as well as disease processes. Let's begin by discussing why the cell cycle is so important. The cell cycle is the fundamental process by which cells grow, replicate their genetic material, and divide. Every living organism depends on this process for development, tissue maintenance, and survival. In healthy cells, division occurs only when it's needed and only after the cell has ensured that its DNA has been accurately replicated. This tight regulation protects the organism from genetic instability. When cell cycle control mechanisms fail, cells may divide in an uncontrolled manner. This loss of regulation is a defining characteristic of cancer and many other proliferative diseases. Because of this, the study of cell cycle regulation is essential not only in basic biology, but also in clinical medicine and cancer research. Many modern cancer therapies are designed specifically to target molecules involved in cell cycle control. And as I mentioned before, I will be discussing forward the major phases of the cell cycle, the checkpoint systems that monitor progression, the key regulatory proteins involved, and the clinical significance of cell cycle dysregulation. Now let me present to you a brief overview of the cell cycle. The cell cycle is divided into two major periods, interphase and M phase. Interphase is the longest portion of the cycle and includes three distinct phases, G1, S, and G2. During interphase, the cell grows, performs its normal metabolic functions, replicates its DNA, and prepares for division. M phase consists of the mitosis and cytokinesis. During mitosis, the nucleus divides, and during cytokinesis, the cytoplasm separates, resulting in two genetically identical daughter cells. Although these phases occur sequentially, progression through the cell cycle is not automatic. The cell must pass multiple checkpoints that verify internal and external conditions before advancing to the next stage. These checkpoints act as quality control systems that maintain genomic integrity. Now let's talk about the four active phases of the cell cycle. The four active phases of the cell cycle are G1, S, G2, and M phase. During the G1 phase, the cell increases in size, synthesizes proteins, and produces organelles necessary for normal function. This phase is especially important because the cell responds to external growth signals and decides whether to continue dividing. Next is the S phase, or synthesis phase. During this phase, DNA replication occurs. Each chromosome is duplicated, ensuring that both daughter cells will receive identical genetic information. Following the S phase is the G2 phase, during which the cell continues to grow and synthesizes proteins required for mitosis. The cell also checks the replicated DNA for damage and makes necessary repairs. Finally, During the M phase, the cell undergoes mitosis and cytokinesis, producing two separate cells, two separate daughter cells. Now, it's important to mention another phase, that is the G0 phase. Not all cells continuously progress through the active phases of the cell cycle. Some cells exit the cycle and enter a resting state known as G0. In this phase, cells remain metabolically active but do not divide. Cells such as neurons and muscle cells often remain in G0 permanently because they are highly specialized and no longer need to divide. Other cells, such as liver cells, can exit G0 and re enter the cell cycle in response to injury or increased demand. The G0 phase highlights the fact that cell division is optional and tightly regulated based on the needs of the organism. As we move on our presentation, let's talk about the cell cycle checkpoints, which are the quality control systems. To maintain accuracy and prevent errors, the cell cycle includes several checkpoints. Checkpoints are surveillance mechanisms that assess whether conditions are appropriate for the cell to proceed to the next phase. They monitor factors such as DNA integrity, cell size, nutrient availability, and chromosome attachment. If a problem is detected, checkpoint activation can pause the cell cycle to allow time for repair. If damage is severe and cannot be corrected, the cell may undergo programmed cell death or apoptosis. The three major checkpoints are G1S checkpoint, G2M checkpoint, and the spindle assembly checkpoint. G1S checkpoint is also known as the restriction point and is one of the most critical control points in the cell cycle. At this checkpoint, the cell evaluates whether it has sufficient nutrients, adequate energy, appropriate growth signals, and intact DNA before committing to DNA replication. If DNA damage is detected, the checkpoint prevents entry into S phase until repairs are completed. This checkpoint plays a crucial role in preventing mutations from being passed on to daughter cells. Loss of proper G1S checkpoint control is a common feature in cancer cells. And then we have the G2M checkpoint that ensures that DNA replication has been completed successfully before the cell enters mitosis. At this stage, the cell checks for DNA damage and verifies that all chromosomes have been fully replicated. If errors are detected, progression into mitosis is delayed until repairs occurs. This checkpoint helps prevent cells with incomplete or damaged DNA from undergoing division, thereby protecting genomic stability. And finally, we get to the spindle assembly checkpoint. And it is in the spindle assembly checkpoint that functions during mitosis. Its role is to ensure that all chromosomes are properly attached to spindle microtubules before chromosome separation occurs. If even one chromosome is not corrected attached, the checkpoint prevents progression into anaphase. This mechanism prevents unequal chromosome distribution, which could result in aneuploidy. Chromosomal instability caused by spindle checkpoint failure is frequently observed in cancer cells. As we move on, let's talk about the cyclins and CDKs, the master controllers. Progression through the cell cycle is driven by two key groups of regulatory proteins, cyclins and cyclin-dependent kinases, or CDKs. Cyclins are regulatory proteins whose concentrations rise and fall during specific phases of the cell cycle. CDKs are enzymes that become active only when they are bound to their corresponding cyclins. Different cyclin CDK complexes operate at different stages of the cell cycle, ensuring that events occur in the correct order. This coordinated interaction functions like a molecular clock, driving the cell toward each phase. Now, there is a regulation for cyclin CDK activity, and cyclin CDK activity is regulated through multiple mechanisms to ensure precision. Cyclin levels are controlled through regulated synthesis and targeted degradation by the ubiquitin proteasome system. CDK inhibitors can bind to cyclin CDK complexes and block their activity, particularly in response to cellular stress or DNA damage. Additionally, phosphorylation and dephosphorylation provide rapid control over CDK activity. Together, these mechanisms ensure orderly and unidirectional progression through the cell cycle. Now let's talk about tumor suppressors and oncogenes in the cell cycle. Tightly regulated cell division depends on a careful balance between proteins that promote cell cycle progression and proteins that inhibit it. Two major classes of genes involved in this balance are tumor suppressor genes and oncogenes. Tumor suppressor proteins function as brakes on the cell cycle. Examples include p53 and retinoblastoma protein, or RB. These proteins monitor DNA integrity and cellular conditions. When DNA damage is detected, tumor suppressors can halt cell cycle progression to allow time for repair. If the damage is too severe to be repaired, these proteins can initiate programmed cell death or apoptosis, as we mentioned before. In contrast, oncogenes function as accelerators of the cell cycle. Normal versions of these genes, called proto-oncogenes, play important roles in promoting cell growth and division. However, when these genes become mutated or overexpressed, they can drive uncontrolled cell proliferation. Examples include cyclin D and CDK4, which push the cell through the G1 phase and into DNA synthesis. Cancer often develops when tumor suppressor genes are inactivated and oncogenes are overactive. This loss of balance removes critical checkpoints and allows cells to divide uncontrollably. Now let's talk about the clinical significance of the cell cycle dysregulation and how it is related to cancer. Cell cycle dysregulation is a central feature of cancer development. One major consequence of checkpoint failure is that cells with damaged DNA are allowed to continue dividing. Over time, this leads to the accumulation of mutations and increased genetic instability. Many cancers arise from mutation in genes that encode cell cycle regulatory proteins, including cyclins, CDKs, tumor suppressors, and checkpoint proteins. These mutations disrupt normal growth control and allow malignant cells to proliferate. Understanding the molecular mechanisms behind cell cycle dysregulation has been critical for cancer research. This knowledge has allowed scientists to identify specific molecular targets for therapy, shifting cancer treatment away from non-specific chemotherapy toward more targeted approaches. As a result, cell cycle regulation is now recognized as one of the most important areas of study in modern oncology. Moving on, I want to talk about the external signals and cell cycle control. The cell cycle does not operate in isolation. Instead, it is strongly influenced by external environmental signals that integrate with internal regulatory mechanisms. One major category of external signals is growth factors. Growth factors are extracellular signaling molecules that bind to receptors on the cell surface and activate intracellular signaling pathways. These pathways stimulate cell cycle entry, particularly during the G1 phase. Growth factors such as epidermal growth factor or EGF and platelet derived growth factor or PDGF are essential for normal cell proliferation. Another important regulatory mechanism is contact inhibition, also known as density-dependent inhibition. In normal tissues, cells stop dividing when they come into contact with neighboring cells. This mechanism prevents overcrowding and maintains tissue structure. Cancer cells often lose contact inhibition, allowing them to continue dividing despite close cell-to-cell contact. Nutrient availability also plays a critical role in cell cycle control. Adequate levels of nutrients, particularly glucose and amino acids, are required for cells to progress through G1. When nutrients are limited, cells may arrest in G1 or exit the cell cycle and enter the G0 phase. Together, these external signals ensure that cells divide only under favorable conditions. Moving on, let's talk about the research methods and experimental approaches. To investigate how the cell cycle is regulated, researchers use a variety of experimental and biochemical techniques. One widely used method is flow cytometry, which measures DNA content in individual cells. By staining DNA with fluorescent dyes, researchers can determine the proportion of cells in G1, S, and G2M phases. This technique is especially useful for comparing normal and cancerous cell populations. Cell synchronization techniques are also commonly used. These methods align cells at the same stage of the cell cycle, allowing researchers to study molecular events as they occur over time. Fluorescent markers and time-lapse microscopy enable visualization of cell cycle proteins in living cells. By tagging proteins such as cyclins and CDKs with fluorescent labels, scientists can observe their location and activity throughout the cell cycle. Additional methods include Western blotting and immunoprecipitation, which detect and quantify specific regulatory proteins, and gene knockout and RNA interference, which allow researchers to study the function of individual genes by disabling or silencing them. These approaches have been essential for advancing our understanding of cell cycle regulation. Moving on, let's talk about the medical significance and treatment approaches. Advances in cell cycle research have led directly to new therapeutic strategies for cancer treatment. One of the most significant developments is the use of CDK inhibitors. Drugs such as palbociclib, ribociclib, and abemaciclib selectively inhibit CDK4 and CDK6, preventing progression from the G1 phase into the S phase. These drugs have been particularly effective in treating hormone receptor positive breast cancer. Compared to traditional chemotherapy, CDK inhibitors are more targeted and often produce fewer side effects, representing a major improvement in cancer treatment. In addition to CDK inhibition, gene therapy approaches are being explored. These strategies aim to restore tumor suppressor function or silence oncogenes, offering potential for more personalized treatment. Another important application is early detection. Biomarkers of cell cycle dysregulation can help identify cancer earlier and monitor response to treatment, improving patient outcomes. The continued development of therapies targeting the cell cycle represents one of the most promising areas in modern oncology. As researchers gain a deeper understanding of cell cycle control mechanisms, new therapeutic targets are being identified. These advances allow for treatments that are more specific, more effective, and less toxic than conventional therapies. Ultimately, cell cycle based therapies have the potential to transform cancer treatment by focusing on the underlying molecular causes of uncontrolled cell division rather than simply targeting rapidly dividing cells. As we are getting to the end of our presentation and class today, I want to present a summary of some key takeaways about what we have presented so far. And I would like to summarize the key takeaways regarding the cell cycle regulation and its importance in biology and medicine. First, cell cycle regulation is essential for life. The ability of cells to grow, replicate their DNA, and divide in a controlled and orderly manner is fundamental to organismal development, tissue maintenance, and repair. Without precise regulation, multicellular organisms would not be able to maintain normal structure or function. Second, cell cycle progression is governed by multiple layers of regulation. These include the coordinated activity of cyclins and cyclin-dependent kinases, the presence of inhibitory proteins, and tightly regulated feedback mechanisms. Together, these systems ensure that cell cycle events occur in the correct sequence and only when conditions are appropriate. Third, checkpoint systems play a critical role as quality control mechanisms. The G1S checkpoint, the G2M checkpoint, and the spindle assembly checkpoint continuously monitor DNA integrity, replication accuracy, and chromosome alignment. These checkpoints prevent damaged or unprepared cells from progressing through the cycle, thereby preserving genomic stability. Fourth, disruption of cell cycle control is a major contributor to disease, particularly cancer. Mutations in genes encoding cyclins, CDKs, tumor suppressors, or checkpoint proteins can remove essential safeguards and allow uncontrolled cell division. This leads to the accumulation of genetic mutations and the development of malignant cells. Finally, understanding cell cycle regulation has significant clinical and therapeutic implications. Advances in this field have led to development of targeted cancer therapies, such as CDK inhibitors, and continue to guide research into early detection, personalized medicine, and novel treatment strategies. In summary, cell cycle regulation represents a central concept in biochemistry that connects molecular mechanisms to cellular behavior and clinical outcomes. By studying how the cell cycle is controlled and what happens when that control is lost, we gain critical insight into both normal biology and the underlying causes of disease. The content presented in this lecture is supported by foundational textbooks and peer-reviewed scientific literature, including Molecular Biology of the Cell, The Cell Cycle Principles of Control, Leninger, Principles of Biochemistry, and the Biology of Cancer. These sources provide comprehensive explanations of cell cycle regulation, its deregulation in cancer, and its therapeutic implications. Thank you very much for your time and attention.